In the ongoing battle against cancer, intrahepatic cholangiocarcinoma stands as a formidable opponent, challenging researchers and clinicians to find more effective treatments. Central to this quest are innovative therapies that target the cancer from multiple angles. Let's explore 4 key agents that form the cornerstone of a promising combination therapy. Gemcitabine. First, we have gemcitabine, a chemotherapy drug that interferes with DNA replication, halting the growth of cancer cells. By integrating into the DNA during the replication process, gemcitabine prevents cancer cells from proliferating, effectively slowing down the disease's progression. Oxaliplatin. Next is oxaliplatin, another chemotherapeutic agent, but with a distinct mechanism. Oxaliplatin forms platinum DNA adducts, leading to DNA cross-linking that the cancer cell cannot repair. This action not only inhibits DNA synthesis, but also triggers cell death, making oxaliplatin a potent ally against rapidly dividing tumor cells. Lenvatinib takes a different approach, targeting the tumor's blood supply. As a tyrosine kinase inhibitor, lenvatinib obstructs the pathways that tumors use to grow blood vessels, a process known as angiogenesis. Without this vital supply line, the tumor's growth is stymied, depriving it of the nutrients and oxygen needed to thrive. Anti-PD-1 antibody. Finally, we have the anti-PD-1 antibody, a form of immunotherapy that empowers the immune system to recognize and attack cancer cells. PD-1 is a protein on the surface of T cells that, when activated, dampens the immune response. Anti-PD-1 antibodies block this interaction, preventing the cancer from evading immune detection and destruction. With this arsenal at our disposal, the study examines the combined efficacy of gemcitabine, oxaliplatin, lenvatinib, and anti-PD-1 antibody in treating intrahepatic cholangiocarcinoma. By analyzing the immune landscape within the tumor, scientists aim to uncover why some patients respond better to treatment than others. Image 1 demonstrates the overall response rate of various therapies for intrahepatic cholangiocarcinoma, with a focus on the GOLP treatment regimen. Image 2 provides a detailed single-cell profiling of the tumor microenvironment during GOLP treatment, showcasing the diversity of cell types and their gene expression. Image 3 highlights the pathways and gene expression changes correlated with iCCA prognosis and GOLP response, offering predictive insights for treatment outcomes. Image 4 shows the impact of macrophage populations on treatment efficacy, linking high macro CD5L+ levels with poorer GOLP response. Image 5 indicates that high baseline levels of cytotoxic CD8 GZMB+ T cells are predictive of a favorable response to GOLP therapy. Image 6 examines TCR clonotype expansion and its influence on GOLP treatment response, emphasizing the role of CD8 T-cell transitions. In part 2 of this video series, I break down the explanation of each image. Image number 1. Image 1 demonstrates the overall response rate of various therapies for intrahepatic cholangiocarcinoma, with a focus on the GOLP treatment regimen. Panel A. Overview of therapeutic efficacy. In Panel A, we see a bar graph comparing the overall response rates of different first-line therapies for intrahepatic cholangiocarcinoma, abbreviated as iCCA. The combination of GMOX with lenvatinib and toripalimab emerges as the most effective, showing promise for future treatment protocols. Panel B. Treatment workflow. Panel B illustrates the treatment regimen for the combination therapy, showcasing the administration cycle over 3 weeks. It details the dosing of gemcitabine, oxaliplatin, lenvatinib, and the anti-PD-1 antibody, providing a clear protocol for medical practitioners. Panel C. MRI results pre and change in tumor marker levels. In Panel D, we have a waterfall plot that displays the percentage change in tumor marker levels from the baseline. Each bar represents a patient, with red indicating a decrease and blue a stable level. This visual aid helps to quickly assess patient responses to the therapy. Panel E. Mouse model injection schedule. Moving to Panel E, we see a schematic of a mouse model study. It depicts the injection schedule of the therapeutic agents and the timeline for tumor measurement, providing a model for how the drug's efficacy is tested before human trials. Panel F. Tumor size comparison. Finally, Panel F shows a scatter plot comparing tumor sizes in mice treated with different combinations of the drugs. Each point represents a tumor, with various treatment groups showing significant reductions in tumor size, supporting the efficacy of the combination therapy. Image number 2. Image 2 provides a detailed single-cell profiling of the tumor microenvironment during GOLP treatment, showcasing the diversity of cell types and their gene expression. Panel A, Research Design and Workflow. Panel A provides an overview of the research design, illustrating the discovery and validation cohorts of the study. It outlines the GOLP treatment regime and the subsequent single-cell transcriptional profiling, highlighting the sophisticated flow of data from treatment to analysis. Panel B, Cell Lineage Visualization. In Panel B, we are presented with a Uniform Manifold Approximation and Projection, or UMAP, which is a technique for dimension reduction. Here it visually distinguishes 12 cell lineages identified in the intrahepatic cholangiocarcinoma microenvironment, color-coded for easy differentiation. Panel C, Marker Gene Expression. Moving to Panel C, we see a dot plot that quantifies the average expression of marker genes across the 12 cell types. Each dot size reflects the percentage of cells expressing the gene, while the color intensity indicates the expression level, offering insights into the cellular makeup of the tumor environment. Panel D, Inter-Cell Type Correlations. Panel D presents a heat map that illustrates the correlation between different cell types within the tumor microenvironment. Redder squares signify stronger positive correlations, and bluer squares indicate negative correlations, shedding light on how different cells may interact within the tumor. Panel E, Cell Type Proportions. In Panel E, we're shown a bar chart that breaks down the proportions of each cell type within the tumor environment, both immune and non-immune cells. This comparison provides a clear depiction of the tumor's immune landscape before and after GOLP treatment. Panels F and G, Gene Expression Correlations. Finally, Panels F and G plot the gene expression correlations before and after GOLP treatment. These graphs help us understand how the treatment affects cellular behavior at the molecular level, potentially identifying targets for enhancing therapeutic efficacy. Image number 3. Image 3 highlights the pathways and gene expression changes correlated with iCCA prognosis and GOLP response, offering predictive insights for treatment outcomes. Panel A, Pathway Enrichment Analysis. Panel A reveals a bar plot illustrating the top enriched pathways of differentially expressed genes before and after GOLP treatment. The green bars indicate pathways significantly associated with a better prognosis when overexpressed pre-treatment, while the orange bars represent those associated post-treatment. This visualization helps us identify potential therapeutic targets and biomarkers for treatment response. Panel B, Gene Expression Scatter Plot. In Panel B, we're presented with a scatter plot contrasting gene expression levels pre- and post-GOLP. Each point represents a gene, with its position indicating the change in expression level and highlighting those genes most affected by the treatment. Panel C, Functional Annotation Clustering. Moving on to Panel C, we see clusters of functional annotations related to the differentially expressed genes, ranked by significance. These clusters provide insights into the biological processes and molecular functions impacted by GOLP treatment, such as PI3K ACT signaling, a key pathway in many cancers. Panel D, Single-Cell Tumor Profiling. Panel D uses a UMAP plot to profile single cells from tumor samples, categorizing them based on their gene expression patterns relative to treatment response. This colorful display segregates cells into distinct groups, helping us appreciate the heterogeneity within the tumor and the varied response to therapy. Panel E, Correlation Matrix. In Panel E, we observe a correlation matrix that examines the relationships between the expression of different genes. The shades of pink and blue denote the strength and direction of these correlations, offering a roadmap of gene interactions within the tumor cells. Panel F, Gene Expression Dot Plot. Panel F features a dot plot indicating the mean expression levels of key genes, as well as the proportion of cells in which they're expressed. The size and color intensity of each dot provide a quick reference to understand which genes are most prominent in the tumor environment. Panel G, Enrichment Scores. Panel G depicts enrichment scores for the gene clusters identified in Panel C. This graph plots the significance of each cluster, allowing us to discern which gene groupings are most relevant to the tumor's response to GOLP treatment. Panel H, Heat Map of Gene Expression. Finally, Panel H presents a heat map detailing normalized gene expression across different clusters. This rich tapestry of color highlights the molecular diversity across the tumor sample and underscores the complexity of the tumor microenvironment, linking high macro CD5L+ levels with poorer GOLP response. Panel A, pathway enrichment in myeloid cells. Panel A showcases a dot plot that represents the pathways significantly enriched in myeloid cells from tumors both before and after GOLP treatment. The size and color of each dot reflect the level of enrichment and the significance of each pathway, indicating potential targets for enhancing treatment response. Panel B, pathways influencing treatment response. In Panel B, we observe the pathways that influence the response to GOLP treatment. This bar graph highlights the differential pathway activation between pre- and post-treatment, providing insights into the biological processes driving the therapy's effectiveness. Panel C, myeloid cell populations. Panel C maps out myeloid cell populations based on the expression of certain marker genes. Each colored dot represents a different cell type, illustrating the diversity within the tumor's immune cell population and its changes following treatment. Panel D, gene expression profiles. Moving to Panel D, a detailed waterfall plot displays the gene expression profiles across various myeloid cells. The plot visualizes how individual genes are up or down regulated, painting a comprehensive picture of the genetic shifts that occur in response to GOLP. Panel E, myeloid cell proportions. In Panel E, we have a box plot contrasting the proportions of all myeloid cells before and after treatment. The plot underscores the significance of certain myeloid cell types in predicting the therapy's success. Panel F, pre- and post-treatment myeloid cell comparison. Panel F features a series of bar graphs, comparing the frequency of specific myeloid cell types before and after treatment. These findings highlight which cells are most affected by the treatment and could be key players in the patient's response. Panel G, cellular landscape shifts. Panel G presents UMAP visualizations capturing shifts in the cellular landscape between patients who responded poorly to GOLP and one patient who showed a partial response, offering a visual correlation with treatment outcomes. Panel H, baseline myeloid cell correlations. In Panel H, we see a scatter plot that correlates the baseline proportion of macro CD5L+ cells with the treatment response, suggesting that higher proportions of these cells might be linked to poorer outcomes. Panel J, clinical correlation with macro CD5L+. Panel J reinforces the clinical correlation, showing that higher levels of macro CD5L+ cells at baseline are associated with a lower rate of response to GOLP, emphasizing their potential role in therapy resistance. Panel K, functional attributes of myeloid cells. Finally, Panel K offers a heat map that delves into the functional attributes of different myeloid cell types, including their roles in inflammation, angiogenesis, and phagocytosis, which could inform how these functions affect the tumor microenvironment and treatment response. Image number 5. Image 5 indicates that high baseline levels of cytotoxic CD8 GZMB+ T cells are predictive of a favorable response to GOLP therapy. Panel A, UMAP of lymphoid subtypes. Panel A presents a UMAP plot displaying the diverse landscape of lymphoid cell subtypes within the tumor microenvironment. This plot categorizes cells into distinct clusters, such as various T cell subsets and NK cells, revealing the complexity of the immune response in the tumor setting. Panel B, heat map of gene expression. In Panel B, we have a heat map illustrating the normalized expression of marker genes across lymphoid subtypes. The range of colors reflects the level of gene activity within each cell type, indicating which markers are most characteristic of each immune cell subset. Panel C, CD4 subtype proportions. Panel C compares the proportions of 3 CD4 T cell subtypes before and after GOLP treatment. This analysis, using paired Wilcoxon tests, highlights shifts in the immune landscape potentially related to treatment response. Panel D, gene expression correlations. Panel D offers a scatter plot that correlates gene expression levels with treatment response. Here, we can discern which genes are most associated with a favorable reaction to the therapy. Panel E, violin plots of PDCD1 expression. Moving to Panel E, we see violin plots showing the distribution of PDCD1 expression, a gene associated with immune cell exhaustion, across different cell types. These plots help us visualize how treatment may affect immune cell function. Panel F, CD8 T-cell subtypes post-treatment. Panel F focuses on CD8 T-cell subtypes post-GOLP treatment, plotting their frequency in the context of treatment response. This data provides insights into the role of these cells in mediating the effects of the therapy. Panel G, correlation between CD8 GZMB+ and patient survival. In Panel G, a correlation graph links the presence of CD8 GZMB+ cells, which are known for their cytotoxic activity, with patient survival. A positive correlation suggests that higher levels of these cells are associated with better outcomes. Panel H, survival analysis. Panel H is a survival analysis graph comparing patients with high versus low levels of CD8 GZMB+ cells. This Kaplan-Meier curve shows a significant difference in survival, underscoring the potential prognostic value of these immune cells. Panel 1, cellular proliferation indicators. Panel 1 presents scatter plots that assess cellular proliferation indicators such as Ki-67, providing a snapshot of how the immune cell's growth dynamics change in response to treatment. Panel K, immunofluorescence of CD8 and GZMB. Panel K displays immunofluorescence images, with CD8 and GZMB proteins labeled, showcasing the presence of these proteins within the tumor tissue. The bright spots indicate the immune cells poised to attack the cancer. Panel L, baseline levels of CD8 GZMB+ and response. Lastly, Panel L shows scatter plots correlating baseline levels of CD8 GZMB+ cells with the treatment response. The plots suggest that higher initial levels of these cells may predict a better response to GOLP treatment. Image number 6 is the last image. Image 6 examines TCR clonotype expansion and its influence on GOLP treatment response, emphasizing the role of CD8 T-cell transitions. Panel A, TCR clonotype ratio. Panel A depicts a scatter plot of T-cell receptor TCR clonotype ratios before and after GOLP treatment. The blue dots represent a decreased ratio, while the red dots indicate an increased ratio post-treatment, hinting at the clonal expansion of T-cell populations. Panel B, CD8 T-cell expansion. In Panel B, we see a box plot showing the expansion of CD8 T-cell clonotypes, classified as contracted, unchanged, or expanded. This helps us understand the diversity and adaptability of the T-cell response following therapy. Panel C, T-cell clonotype classification. Panel C provides a bar graph categorizing TCR clonotypes based on their contraction or expansion status. This classification may be crucial for evaluating the immune system's adaptability to the therapeutic regimen. Panel D, clonotype expansion and treatment response. Moving to Panel D, a box plot correlates clonotype expansion with treatment response. It shows that patients with a partial response exhibit significant T-cell clonotype expansion, suggesting a link between immune activation and therapeutic success. Panel E, clonotype proportions. In Panel E, another box plot compares the proportions of specific TCR clonotypes before and after treatment. The variation in these proportions can give insights into how treatment influences T-cell populations. Panel F, CD8 GZMB+ and response. Panel F highlights a correlation between CD8 GZMB+ cells and treatment response. The graph indicates that a higher presence of these cytotoxic cells correlates with a better response, underscoring their importance in tumor control. Panel G, CD8 T-cell subtype shifts. Panel G presents box plots comparing the shifts in CD8 T-cell subtypes between patients with stable disease and those with a partial response, providing evidence of the dynamic changes within the CD8+ T-cell compartment in reaction to treatment. Panel H, interclonotype correlations. In Panel H, we're shown a heat map of interclonotype correlations. The color intensity indicates the strength of the relationship between different T-cell populations, offering a snapshot of the immune cell interactions. Panel 1, UMAP of CD8 T-cell states. Panel I uses UMAP to visualize the transition states of CD8 T-cells, from proliferating to cytotoxic to exhausted. The different colors represent various functional states of these cells, reflecting their roles in the immune response to the tumor. Panel J, pseudotime trajectories. Panel J presents pseudo-time trajectories illustrating the developmental pathways of T cells from a naive or proliferative state towards exhaustion, providing a temporal view of T cell dynamics. Panel K, clonotype status shift. In Panel K, a violin plot reveals the changes in clonotype status from pre to post-treatment, offering a closer look at how individual T cell clones respond to the therapy. Panel L, computational modeling of clonotype shifts. Panel L depicts computational modeling of clonotype shifts, comparing the states of T cell populations before and after treatment. This analysis helps in understanding the effect of the therapy on T cell behavior. Panel M, survival analysis. Moving to Panel M, a survival curve relates T cell clonotype status to patient outcomes. The graph suggests that specific T cell clonal shifts might be predictive of survival, emphasizing their potential prognostic value. Panel N, mutation ratios. Panel N offers box plots that show mutation ratios in patients with different treatment responses, indicating that T cell clonotype status might influence or be influenced by tumor mutations. Panel P, clonotype and treatment response. Finally, Panel P correlates specific T cell clonotypes with the treatment response, reinforcing the concept that certain immune profiles are more conducive to positive outcomes. If you found this breakdown helpful and would like to see more content that demystifies complex research for the general public, researchers and patients alike, please consider subscribing and giving this video a like. Your support enables us to continue producing content that advances understanding and fosters an informed community. For those interested in delving deeper into the study, we've included a link to the full publication on PubMed in the YouTube description below. This resource is invaluable for further study and reference, and we encourage scholars and curious minds to explore the detailed findings and cite them in their own work.